A very good afternoon to you all. I am Dr. Lubna Sethi, and I welcome you with immense pleasure on behalf of the Department of Pharmacology and Therapeutics to commence the Pharmacovigilance and Prescription Audit Program at our prestigious King George's Medical University, Lucknow. I would like to welcome the dignitaries seated on the dais. Our Honorable Vice Chancellor, Sir, Lieutenant General Dr. Vipin Puri, Sir. A very warm welcome, sir. Professor Y.P. Gupta, sir, the guest speaker for today's program, President of All India Institute of Medical Sciences, Bhopal, and President of All India Medical Sciences, Jammu. It is an honor to have you here, sir. Heartiest welcome. Dr. Jitendra Verma, sir, CEO of Life Care Innovation. Most welcome, sir. I would also like to welcome the guests who are participating online with us today. Dr. Sunil Kumar, Director General, Health Services, Ministry of Health and Family Welfare, Government of India. I welcome you, sir. Dr. Jay Prakash, Principal Scientist, Indian Pharmacopoeia Commission, Ministry of Health and Family Welfare, Government of India. A very warm welcome, sir. Dr. Pooja Gupta, Coordinator, All India Institute of Medical Sciences, Pharmacovigilance and Material Vigilance Program, Ministry of Health and Family Welfare, Government of India. Heartiest welcome, ma'am. I also welcome Professor Amol Kumar Sachan, sir, our Head of the Department of Pharmacology and Therapeutics, King George's Medical University, Lucknow. I will now invite Dr. Amol Kumar Sachan, Professor and Head, Department of Pharmacology and Therapeutics, King George's Medical University, to welcome the guests and introduce Professor Y.P. Gupta, sir, the guest speaker for today's program.
Good afternoon, sir. Good afternoon, sir. Today, we have assembled here for a guest lecture on pharmacovigilance and prescription audit. This program is a long-awaited program. In fact, the importance of the pharmacovigilance has increased tremendously this time after the COVID pandemic. We have used many drugs empirically, the effect of which were not known previously, but when that disease has come all over the world, we have used many drugs. And because of that, many complications, many adverse effects have appeared. The mucormycosis is one of them, which has really affected our country among most of the developing countries. In Western world, it has not been seen so much. And thereafter, the importance of the pharmacovigilance has increased. Besides that, there are various drugs which we commonly use, and because of various reasons. For example, there is a drug-to-drug interaction, there is a side effect of the drugs, there are various drug disease impact. Because of that, there are irrational prescription writing. The generic drugs which commonly should be used as emphasized by the government, and here also by our Honorable Vice Chancellor is emphasizing that we should use generic drugs. Besides that, we should also emphasize on pharmacoeconomics. The drugs should be economical. They should be not only from a better company, they should be effective, but they should also be quite economical to the patients. And that's how these two domains, they have taken an important role in our day-to-day practice in the clinical departments. We are very thankful to our Honorable Vice Chancellor, Lieutenant General Dr. Vipin Puri, for giving his guidance and support to initiate these programs in the university. He has already issued circulars to start these programs, and the detail of those working of these pharmacovigilance and prescription audit will be discussing subsequently in a few weeks' time with the nodal officers in the different clinical departments, and will formulate how to collect the data for pharmacovigilance as well as for prescription audit. I would like to welcome all the dignitaries who are sitting in the dais, particularly Professor Y.K. Gupta, who has come long way from Delhi by road. He has taken a lot of time. He has started today at 6 a.m. and he has just reached the college just now. I am very thankful to him as he has always inspired us. Many of you might not be knowing that he belongs to this institution. He has graduated and post-graduated from this institution, and because of this affinity towards this institution, he is all the time ready. Whenever we call him, he is ready to support in any whatever the best possible manner, and I'm really obliged and thankful for all that cooperation which he has extended. I would also like to welcome Dr. Jitendra Verma, the CEO of the Life Care Innovation. However, there are many pharmaceutical companies in the country, but incidentally, after the development of this mucormycosis as a serious menace in our hospital as well as all over the UP and India, his company has really contributed a lot. He is producing lyophilized amphotericin B, and many of you might not be knowing the production of this lyophilized amphotericin B is here in Biotech Park in Lucknow. I am thankful for Dr. J.S. Verma that he has spared the time. He has also come from Gurgaon. I would like to welcome Dr. Jay Prakash, who is the principal scientist in the Indian Pharmacopoeia Commission. Dr. Pooja Gupta, who is the coordinator of Pharmacovigilance and Vigilance Program, Minister of Health and Family Welfare, Government of India, and she is working in the All India Institute of Medical Sciences in the Department of Pharmacology, who is supposed to be connected. Anyhow, she is having some engagement up to 4 o'clock. I have requested her that if she can come for even for some time, she will join with us. I would also like to welcome Dr. Sunil Kumar, who is the Director General Medical Health, who should join after some time. Dr. Y.K. Gupta is an alumni of our department. He has been pioneer in the field of pharmacovigilance. I'm very grateful that he has come today during this COVID time. Also, he has taken sufficient time for us. He has held very prestigious position. Like currently, he is the president of All India Institute of Medical Sciences, Bhopal, as well as the AIMS is coming in Jammu. He is also president of that AIMS. He has been head of the Department of Pharmacology in All India Institute of Medical Sciences and also former dean of the AIMS Delhi. In charge of the National Poison Information Center, National Scientific Coordinator of Pharmacovigilance Program of India, former director, CSIR. He has worked here for 3 years in the ITRC, previously it was known as ITRC. He has remained as a director there and former president of Indian Society of Nanomedicine, former president, Indian Pharmacology Society, principal advisor, Indian Strategy Development, chairman, board of directors, PvPI and DBT and Government of India, member, general body and governing body of the National Lab, Government of India, member, governing body, the National Institute of Immunology, DBT, Government of India, member, governing body, Regional Institute of Biotechnology, DBT, Government of India, vice chairman, Standing National Committee on Medicines and Healthcare Products, Government of India, chairman, National GLP Technical Committee, BST, Government of India, member National Committee for the International Sciences, Council of India, INSA, and President National Society of Alternatives to Animals. He has been operational everywhere where there was a drug. The Dr. Y.K. Gupta is there. So whenever we have got anything related with the drugs for which we are there, he is always helpful to us. We are, as you know, we are starting this prescription audit program and this pharmacovigilance, and Dr. Y.K. Gupta has been instrumental to get these permissions from Government of India. We are going to start this prescription audit program with the help of ICMR shortly. We are going to apply to the ICMR. They start a center, and we will be having this center here in our university. Pharmacovigilance center already been allotted to us, and shortly we will have that program effective within a few weeks' time. Now I invite Professor Y.K. Gupta to deliver his lecture. Thank you very much.
Adharaniya Lieutenant General Vipin Puri, Vice Chancellor of this prestigious King George Medical University, Dr. Amol Sachan, Head of Department of Pharmacology, Dr. Jiten Verma, the speaker of this, and I see my friend, I must mention his name first, Dr. Chandishwar Nath, my batchmate, and not only my batchmate here, but in my formative age, my guru also. And also Dr. Jagdishwar Sahai, I can see we have been here for, I think since we were working for entering into MBBS, that was class 12 until now. And I can see Dr. Srivastav, and I can see many of those familiar faces. But more important is this familiar hall, this familiar building, this familiar surrounding, this familiar, what you can say, smell, fragrance of this institution, this college, which is unique in the world. Whether you go in Delhi, whether you go in Tokyo, whether you go any part of the world, if any person from Lucknow visits that place or meet you, more so if he has a connection with KG Medical College, you feel such a fresh air, you feel such a nostalgic era where you are entering into, you will never realize that what it is. And I was just talking today to Dr. Balram, who is now DG ICMR, and who is incidentally son of Professor K.P. Bhargav, who used. And I said कि यार हम जा रहे हैं KGMC में आज. I said कि कितने बजे निकल रहे हैं आप? He was so keen कि क्या मैं भी चलूं आपके साथ में? मैंने कहा हम तो already हम इसमें expressway पे आ गए. That is what the feeling people have wherever you reach, whether you are in a top position or you are working in a remote village for the public, you have that much of feeling. So I'm not giving this as a lecture, Sachin, but I am just maybe just like to interact with it. And I was just saying, Dr. Puri, my association with the KG Medical College is not from my duration, but my grandfather, whose name is still here, Dr. H.N. Gupta, 1925 Dept medalist here. And he was always so proud when I was here a student. He used to say my pet name, I will not name it. जाके देखना है, जाके वो जगह है कि नहीं, वहाँ पे physiology department में उससे कम. That type of memory he still used to carry when he died at age of 99. He was just short of 1 year. He was always saying, "मुझे 100 साल पूरे करने." and used to be invited as their chief guest in Georgians meet as the eldest person living, surviving at that time, that area. So, and I will also acknowledge the Department of Pharmacology here, where I did my MD in 78. And we remember those days when we used to run after the dogs to catch it, because we were working on vomiting experiment with Professor PvPI, and एक कुत्ता पकड़ के ले आए तो that was the victory for us, used to be. सड़क पे से झम्मन लाल के साथ में हम जाते थे. And then we used to celebrate that, myself, Dr. Chandishanath and all those things. And that's why I'm saying so, because when Dr. Sachand says, yes sir, आप यहाँ पे आइए, I said, यार तुम्हें कहने की जरूरत नहीं है, just हमको बहाना चाहिए यहाँ पे आने का. And I remember so many days जबकि यहाँ पे उस समय कल्चरल प्रोग्राम होता था, और एक बार प्रोफेसर पीसी दुबे एक थोड़ा सा मॉडर्न जमाने का डांस किसी स्टूडेंट ने किया था, थोड़ा सा, जो कि आजकल तो बहुत ही ज्यादा शालीन माना जाएगा, और वो उठ के चले गए. उस समय का वो फंक्शन बंद हो गया. That was the era. That was the era when we used to sit here, and this hall where we are sitting here is a tremendous memory of that. And I'm fortunate to be invited here. I've traveled a lot when I passed out from this college, and then was director at CSIR and also in IITR, and sometimes as a functioning director of CDRI, then in the governing council of these institutions. And during the process, what I realized that pharmacology is a discipline which should not remain in the captivity of the building. The real pharmacology comes when you, outside word, you shake the physician who prescribes medicine. You shake the ministry who creates the policy. You shake the economist who make the policy decision कि ये दवाई इतने की बिकनी चाहिए. And that's why I chair the committee by PvPI and direct them that how and what policy should we make to make the drugs affordable for the country. You shake those people who make the policy for hospital, for insurance. And that's why I am what is called as a now Standing National Committee on Medicine, which makes the essential list I'm going to release in the next 3 days. And you are the group which should shake the Medical Council of India that ये drugs पढ़ाने का तरीका अब बदल दीजिए. ये नहीं कि हम जो कह रहे हैं कि अब साइड इफेक्ट बताओ, हमने कहा एक और बताओ, लड़का बताता गया, फिर बाद में हम पूछते हैं नहीं वो जो मेरे दिमाग में है वो जब तक नहीं बताओगे पास नहीं होगा. अरे यार वो जमाने खत्म. Now you have to be relevant today, and the prescribers have to be relevant. When a prescriber, whether he is a surgeon, physician, pediatrician, or those who are involved in the policy of purchasing drug, unless they consider the risk of this drug, unless they consider the relative benefit of this drug, unless they consider what is the economics involved in this drug, till that time, the budget resource country like India will not be able to take this drug to the public, which is not sitting in Lucknow or Delhi, but we are looking at the people who are sitting in Ramnagar, Budwal, Barabanki, where I did my class 10th, a village. That is important, and that is the purpose, I would say, our national list of essential medicine. We have 6,000 plus medicine today. What we have created a list of 347 drugs which are essential and which can treat 80% of the country's population. Why a normal physician in an ordinary situation, whether it is a primary care, secondary care, tertiary care, should go beyond that? And if you are going beyond that,
you have to give justification for that. If the drug, when you say pharmacoeconomic principle, if you are giving a drug to a person which is a patient in 7 days and costing about 600 rupees, what is the justification for giving a drug or drugs which will cost him 6,000 rupees? Because you have not learned it. Because the education which you have got was 10 years or 20 years back in classes, during internship. But we never did this early level. And that's why we started skill-based learning in MCI. A group of us met here, it has to be relevant to the teaching. And that's why you have now skill-based learning. And I was involved in developing drafting of the skill-based learning in medical council. Now, shifting this, we started a program, what is called as this. In 2010, we started at All India Institute of Medical Sciences as a small program. That is, you collate, and at that time, when I used to conduct meeting of Pharmacovigilance program, PvPI, my wife used to say, where are you going in this meeting at this time? Again and again, ये Pharmacovigilance, ये vigilance तो storekeeper लोगों को check करने के लिए होता है. Why you are involved in this? That was the concept at that time. And that was the response of my wife. She was a professor of radiotherapy. The ignorance of the pharmacovigilance, vigilance karo. Not that the adverse drug reaction which is happening, and then you have to keep a strict vigil on that. Because unless you know that this drug can cause this side effect, and unless you know this drug can cause side effect in Lucknow, the same side effect is seen in Bombay, the same side effect is seen in other places in the world, and you collate them, and then you do the risk-benefit analysis, and after this risk-based analysis, and you do the causality assessment, then you make a case to the government that this drug should be banned in India. Before that, even today, if you just see the drug ban hoti hai, drug kahan se ban hoti hai? Because the data which is coming is from a Scandinavian country or from UK, which is only 2% or 3% of the world's population, whereas this 40% population of the world is not able to contribute the data for that. Matter of shame at that time. And you must realize that in spite of 60, 70 years of our independence when we started this program 10 years back, the WHO-sponsored Uppsala Monitoring Center used to charge $10,000 from us to put our adverse drug reaction data into their program. And then, उसी data को mature करके, उसी data को बना करके हमको वो बेच देते हैं कि आपकी ये एस्प्रिन में, पैरासिटामोल में 20% लोगों में लिवर डिजीज होती है, 10% में ये होती है, और उसके बाद में हम कहते हैं यार टॉक्सिक है या ये सेफ है. ये वही बात थी कि हमसे सूट खरीदा, हमको कपड़ा बेचा 4 गुने दाम पे. And now when we started this program, now this program has 400 centers across the country. And from our aims, this is Indian Pharmacopoeia Commission, and Dr. Jayaprakash is Chief Scientific Officer of that, and I am still continuing as the Principal Scientific Coordinator. Now that same WHO is now paying us because we are putting this data into their portal. Now they're paying us. Now see the importance of the data, and you see the importance of collection, collation, and analyzing this data system. Important thing here is, अगर आपने कोई या क्लिनिक है या अस्पताल में किसी का एडवर्स ड्रग रिएक्शन देखा, what you do? You remember it. अब बाद में कहे मैंने भी ये एक एडवर्स ड्रग रिएक्शन देखा, उसको बड़ी एलर्जी हुई थी. लेकिन ये डेटम प्लूरल नहीं बन पाया. जब डेटम से डेटा बना तो भी कोई फायदा नहीं है. और जब ये डेटा जब इंफॉर्मेशन बना कई जगह से कलेक्ट होकर जर्नल में से आके हमने बनाया, तब भी यूजलेस है.
जब तक कि ये डेटा इंफॉर्मेशन से कन्वर्ट होकर नॉलेज में नहीं आएगा, and then we say you translate this knowledge, the physicians will not translate this knowledge, that yes, this Steven Johnson syndrome can happen, carbamazepine, this dose, and therefore you should be cautious while giving this drug or this medication in this particular population. And agar ho jata hai, then how to handle it. Risk identification is important. Risk mitigation is also important, and risk assessment is important. And that's why you see the new disciplines have developed. Now we have a CHIPS, and Department of Biotechnology, based on our recommendation, has given a thrust, and this is for information to all. If you can develop a genome-based or genomic analysis, if this gene is involved in this particular side effect, and that's why you see in anti-cancer drugs, we have a genomic analysis. And this person, this group of patients should not be given this biosimilar because this population has this gene, and therefore the side effects will be much more, and this side effect or this selection of this anti-cancer drugs will be better in this situation. So patient-based selection of the drugs is now possible. That's the pharmacovigilance. If bedaquiline is not effective, if artemisinin is not effective in certain population, what do you say? You will say कि यार ये तो बड़ा गड़बड़ है, bedaquiline से resistance develop हो गया. No, you have to go into this. That is it because of the compliance? Is it because of the poor drug? It is because of the quality, and then you say, is it because of the genomic disturbance, this distribution changes? And that is what is required. And that can happen only when the data is generated from different parts of the world, different parts of the country. And that's what I have been persistently telling Department of Pharmacology, that it is unfortunate that the entire country is knit now into pharmacovigilance program. But it was really a pain in my heart sometimes कि जहां से हम निकले हैं वहां पे ये भी सेंटर भी नहीं बना पा रहे. And I am thankful to Dr. Sachan and the Vice Chancellor that the group which should have grabbed it 20 years, 10 years back is at least now willing to take it. And similarly, today the relevance has been realized by the word of Pharmacovigilance Program of India. This is now the world's largest pharmacovigilance program of India, where MCI has made mandatory pharmacovigilance program of India. And we have a 450 plus pharmacovigilance centers. We are taking now the decisions on the drug withdrawal, changes in the drugs indication, precaution based on our data. So we are now, we can say, हम आत्मनिर्भर हैं अपनी दवाइयों पे निर्णय लेने के लिए अपने डेटा से. And this can happen only when the entire IT system is effective. The other thing which I would also say is everybody prescribes medicine. But you see the type of prescription are generated, the type of medicines which are prescribed. So many errors, so many deaths are occurring because of the handwriting of the physicians is so good. I would say, quote, that only maybe even wife cannot read it, maybe girlfriend can read it. The physician's handwriting can only be read by a particular pharmacist. So many cases of Folvite, and I was one of the inquiry committee where Folvite was prescribed, intended to be prescribed by a physician at All India Institute of Medical Sciences, and he was given methotrexate as Foltrexate brand name. And the patient took this for 6 months and came at the terminal stage of kidney disease. And then inquiry was set up. Whose mistake? The physician mistake. Physician ने कहा यार हम तो यही writing है, Folvite लिखा, Foltrexate कहाँ से पढ़ दिया? केमिस्ट कहता है साहब ये तो बिल्कुल Foltrexate लिखा है, हम तो 6 महीने से रिपीट प्रिस्क्रिप्शन देते जा रहे हैं. The problem is गांधी जी रिपेंटेड थ्रू आउट हिज लाइफ कि मेरी हैंडराइटिंग खराब थी। हमने गांधी जी की वो सेंटेंस नहीं सुना, हमने कहा गांधी जी की राइटिंग जैसी मेरी राइटिंग है। गांधी जी रिपेंटेड ऑलवेज कि his bad handwriting, and therefore, and he prescribed so much. You remember, you know, the tuberculosis, resistance of tuberculosis, major cause of in early days developing resistance of tuberculosis was inadequate prescription writing. Because now people write 6 months लो 1.5 साल तक लेना है। उन्होंने कहा ठीक है, जब तक खांसी आए फिर मेरे पास आना, 2 महीने बाद फिर मेरे पास आना, जब तक तुम्हारा बुखार है। उसने दवाई ली, because of the economic situation was not there, DOT program was not there, so program started after that when people realized कि लोग तो बीच में छोड़ देते हैं, और उसके बाद that became the reason of the resistance. The patient compliance is important. You prescribe a medicine which is expensive is important. And that is why we realized, even after so many years of this, the precipitation of this has happened when the COVID happened. And this was realized that we are treating COVID as a pandemic, but we are not realizing the underground pandemic of antimicrobial resistance. So many antibiotics, whether it is azithromycin, whether it's a broad spectrum or cephalosporins, randomly prescribed by the physicians. अगर प्रिस्क्राइब पेशेंट को भी ना करो तो क्या साहब एजिथ्रोमाइसिन तो आपने बताई नहीं थी, लेकिन हमारे पड़ोसी ने बताई तो हम खा रहे हैं। Even today, when so much of education through TV we did, people are prescribing antibiotic, realizing that antibiotic has no role in viral. Only on the background कि अगर secondary infection हो जाए तो उसको prevent कर ले। अरे भाई होने तो दो, कोई indication हो तब तो दो, not that blind. Then that is causing the major concern, and that has resulted into the Ministry of Health giving this direction that all prescription audit of cancer of COVID patient must be done. The cause of mucormycosis or bacterial or the fungal infection or opportunistic infection, because of perhaps the legitimate, only legitimate indication in COVID drugs is corticosteroid, but the overuse of corticosteroid, untimely use of corticosteroid, and without realizing that which patient should not be given or should be given with caution, corticosteroid has resulted into fungal infection and several deaths of mucormycosis. The corollary of that, we were not prepared to give the correct drug for mucormycosis. Whether it is amphotericin, whether it is amphotericin osteolate, or whether it is a amphotericin, liposomal amphotericin, which is kidney safe, relative kidney safe. Now, the important thing is we say relative kidney safe. Do we have the data of all the COVID? This was the golden opportunity for us to collect the data of how many people had a kidney problem. And can we give some additional kidney protective agent, whether it is a 1000 दवाइयां हमारे पास, अगर आप देखें आयुर्वेद में कहते हैं कि kidney protection, kidney never protective, liver protective. And this was the opportunity where you could have tried, you could have done trial कि kidney protective drug हम दे रहे हैं और उससे हमारा side effect नहीं हो रहा, बच रहा है. And that's why ICMR started this. You must have seen we have included this in the call for proposal for ICMR, different clinical trial of these small things. Now, if we know that what is the correct dose in Indian population of steroids for how many days, at what time, perhaps if at all the third wave comes, we will be better prepared. We found that there is a sudden shortage of amphotericin or all the drugs, whether it was any of the drugs which had been used as emergency use authorization. Under this, सारी दवाइयां बाजार में से गायब हो गई, and या फिर जो दवाई 500 रुपए की थी वो 5000 में आने लगी, और जो 5000 का वेंटिलेटर था वो 50000 का हो गया, ऑक्सीजनेटर जो 10000 का था वो 1 लाख तक पहुंच गया, because this was a stateless state at that time. And why? Because we created this situation. We did not anticipate that. And therefore, the need of this gathering, this institution, which is the leader, I would say, KG Medical Institution is the leader institution of the medical sciences. Why we not make a pragmatic, a possible, predictive indication that अगर ये होता है तो इतनी बेड्स की जरूरत होनी चाहिए, अगर इतना होता है तो हमारे पास इतना बफर एम्फोटेरेसिन लाइपोसोमल का होना चाहिए. ये 6 महीने की या 1 महीने की जो हाफ लाइफ है, इसको हम 2 साल की क्यों ना करें? This is the out of box thinking which is people have started thinking now. What is called as this is the necessity, is the mother of invention. But the wise people are saying that when they anticipate the necessity, and that is why intellectual things are required. I am very happy that, and I will just say, if those who are here, हमारे पास एम्फोटेरेसिन की शॉर्टेज आई, एंड आई वाज पार्ट ऑफ दैट कमेटी के. इसको कैसे करें? गवर्नमेंट ने स्टेट को बांट दिया कि अब तुम कंट्रोल पे ले लो, एम्फोटेरेसिन पे लाओ, जो जितने मरीज हैं उसके हिसाब से हम इनको एम्फोटेरेसिन लाइपोसोमल एम्फोटेरेसिन देंगे. लाइपोसोमल एम्फोटेरेसिन की कमी हो गई तो हमने कहा जी अब दूसरा एम्फोटेरेसिन भी लगा दो विद XYZ प्रिकॉशन. फाइन, बट वी डिड नॉट टेल, परहैप्स वी थॉट दैट इट इज अंडरस्टूड बाय द फिजिशियन कि वो किडनी फंक्शन को देखेंगे ठीक से. नाउ यू हैव टू बैलेंस इट, द रिस्क बेनिफिट इकोनॉमिक्स, and that is what is important to us. And that's why we have, and very happy that we have offered this therapeutic audit also as a center, which is now 9 centers are doing it. And Dr. Sachan, जिस दिन मैंने उनसे कहा, I think he took approval from Vice Chancellor, and immediately he sent about 100 prescription of COVID patients, which have become a part of the pool now. They are working together. Again, my thing is, these are the issues of national importance, national issues. We are talking of drug pricing today. Input has to come from us. And unless this input comes from this institutions like this, I think things will be slow. And then we will be only saying, यार ऐसा होता है, हम फिर कहते हैं कि सरकार प्रिपेयर्ड नहीं थी. अरे हमने सरकार को प्रिपेयर करने के लिए अपना इनपुट दिया. हमारी रिस्पॉन्सिबिलिटी नहीं थी एज ए इकोनॉमिस्ट, एज ए फार्माकोलॉजिस्ट, एज ए एपिडेमियोलॉजिस्ट, that we could have predicted better कि हमें इतने की जरूरत है, इतनी बेड्स की जरूरत है. और अभी भी हम नहीं बता पा रहे कि कितनी बेड्स को हम रिजर्व में रखें, कितनी बेड्स को हम रीयूटिलाइज करें, कितनी को हम ऑप्टिमाइज कैसे करें. I think that is what group dynamics or group discussion need to be. So I am extremely thankful again that VC sir and all of you are here, and we will interact more and more, but I would like my alma mater to be leader in all national policy making issues. Thank you very much.
Thank you so much, sir, for sharing your memories and your knowledge with us. And we are so honored by your presence today. I would now request our Honorable Vice Chancellor, sir, Lieutenant General Dr. Dept, sir, to address the audience and share with us some words of wisdom.
Professor Y.K. Gupta, Vice Chairman of the Standing National Committee on Medicines and Healthcare Products, Government of India, and also the President of AIIMS at Bhopal and AIIMS Jammu, Professor Sunil Kumar, Director General, Health Services, Ministry of Health and Family Welfare, Professor Jay Prakash, Principal Scientific Officer, Indian Pharmacopoeia Commission, Dr. Pooja Gupta, Coordinator of the Pharmacovigilance and Material Vigilance Center at AIMS, Jitendra Verma, CEO of Life Care Innovation, Professor Sachan, Head of Department Pharmacology at King George Medical University, faculty, deans, residents, ladies and gentlemen. At the outset, let me compliment the Department of Pharmacology, the very dynamic Professor A.K. Sachan, for organizing this very important seminar on pharmacovigilance and prescription audit. Let me begin with by telling Professor Y.K. Gupta that the audience that you see here today, sir, is the heads of departments of this very prestigious university. But 3 times or 4 times a number have watched you and heard you on the online platform. So this is the interest that your talk has generated in this university. So that was compliments to the Department of Pharmacology and compliments to Professor Gupta, an alumnus of this college, an alumnus of this university. We are so proud to have you today with us this afternoon. Well, friends, ladies and gentlemen, let me begin by saying we have already flagged he's flagged such very important contemporary real-time issues in today's times, which I feel perhaps we have been left behind. This college has perhaps been left behind in this race, in this monitoring, this policing of the drug industry. And I think with increasing utilization of medicines, drugs, which are indicated and sometimes not indicated, use of prescriptions well written, not so well written, drug abuse in its varied dimensions, and of course, documentation of adverse drug reactions. Now, this is a conglomerate of problems, issues, challenges which face us as far as we as healthcare professionals look at this problem on a larger page. And I think it's very importantly flagged by Professor Gupta. In fact, in some countries, adverse drug reactions is amongst the top 10 leading causes of death. Now that's something which is amazing. And I think that's the point we have to register. We tend to push it under the carpet, but I think there is a need for us to understand this. And I think therefore, the concept of drug safety pharmacovigilance has evolved considerably in the recent past. And as Professor Gupta says, now there are more than 450 centers all over the country trying to collate and this data. And now the WHO is asking us for this data. And we are being paid for this service because of the volumes that we generate, we make such important information for the world at large, and therefore the importance of pharmacovigilance. I actually acknowledge that the Medical Council of India has made it mandatory that every medical college today in the country should have a pharmacovigilance committee as per the current regulations. So towards that extent, the KGMU has decided to run its own adverse diverse drug reaction committees, and I'm sure the Department of Pharmacology will take it to its logical conclusion in the coming days and weeks. Inappropriate use of drugs is an irrational prescription writing. These are some other issues which come to mind, and I am flagging this in this open forum because now this is not only unique to this college, unique to various medical colleges in India, but unique to all healthcare professions all over the world, that inappropriate, irrational prescription writing has become an order of the day. And therefore, there is a need for us to do some sort of an audit of prescriptions and try and understand why and what and how, and ask some very important introspective questions. Well, I want all of you not to go away with the impression that we're trying to do policing of prescriptions. No, it's not that. It is trying to put things into perspective. It's trying to rationalize prescriptions, why and how, and answer those questions. Sometimes one tends to get carried away by the industry, the industry tries to push you into decision making. It is for you to put into perspective that what you write has got huge implications. Some issues were raised about pharmacoeconomics, but I say pharmacovigilance is as important. They're all interrelated matters, whether you call it economics, whether you call it vigilance. The side effects, the adverses that you are creating or you may create, the drug interactions that you might create by so-called polypharmacy that we keep talking about. I think those are some of the issues I'm flagging which require to be looked at when we do prescription audits. And of course, I would want my Department of Pharmacology to look at all of these. So once again, with these few words, I thank our guest speaker this afternoon. I thank the veteran pharmacologists who are also here this afternoon, with deep respect and regards to you also. It is a pleasure for me to be a part of this very important webinar and seminar this afternoon. Thank you, ladies and gentlemen.
Thank you for your visionary words and guidance, and for your continued support towards our department. Thank you, sir. Now I would invite Dr. Jay Prakash. He has joined us online here today. He is the Principal Scientist, Indian Pharmacopoeia Commission, to share her words on how to go for COVID-19 pharmacovigilance.
Thank you, madam. Am I audible? Am I audible? Am I audible?
Yes, sir.
Yes.
Yes,
Good afternoon, everyone. Honorable Vice Chancellor, King George Medical University, Lieutenant General Professor Vipin Puri, Professor Y.K. Gupta, former Head of the Department of Pharmacology, All India Institute of Medical Sciences, and President AIMS Jammu and AIMS Bhopal, Professor A.K. Sachan, the Head of the Department of Pharmacology at King George Medical University. I am very grateful to Professor Y.K. Gupta and all the distinguished personals of King George Medical University for inviting me to this important event about the COVID-19, the pharmacovigilance of COVID-19 and prescription audit. So I have a few slides to present, and I think you will find the interesting data from the Pharmacovigilance Program of India. So I will just give you about the mandate of the Indian Pharmacopoeia Commission. So we publish the Indian Pharmacopoeia, which is a legally recognized book of standards for medicines in our country. And on 31st of March, IP Addendum 2021 has been released. We also provide the reference standards for the compliance of the monograph set out in Indian Pharmacopoeia, and Ministry has also given the mandate for timely publication of National Formulary of India, which is a guidance document for healthcare professionals for rational use of medicines. And here I would like to put the gratefulness to my guru, Professor Y.K. Gupta sir, who has been instrumental in giving the guidance, support, and the time more than required for continuous update of the National Formulary of India. And we are in the process of getting the current edition of National Formulary of India 2021 to be released in next month. And we also run the Pharmacovigilance Program of India, and the credit of supervising this program and giving the direction to the program goes to Professor Y.K. Gupta and all the team members who have been dedicatedly and deeply associated with Pharmacovigilance Program of India. And the KGMC being one of the premier institutions in the country and one of the prospective centers under PvPI, I would be requesting them to provide us the support for the PvPI as well as the other activities in which their crucial role will be very beneficial for each of us. So, sir has already mentioned about how the pharmacovigilance of drugs is important, and today it not only covers the scope of drugs, but in its scope we have the drugs of variety of origins such as chemical origin, the biological origin drugs, medical devices, and the scope is beyond the medical devices also. So I remember the time that sir, under his guidance, the 2 days workshop was organized at All India Institute of Medical Sciences in 2010, and from there it had been operationalized. So the program, the National Coordination Center had been at All India Institute of Medical Sciences under the guidance of Professor Y.K. Gupta. So till 15th of April 2011, and after that the center had been running from Indian Pharmacopoeia Commission at Ghaziabad. So since then, under his constant supervision and guidance, we have been able to expand this program, and today you can see that there are 395 adverse drug reaction monitoring centers throughout the country, and they have been increased in a phase-wise manner. If you look at the reporting status of the adverse drug reactions, so this slide shows the trend of the reporting from 2011 to 2021 onwards, and till date, more than 5 lakhs of individual case safety reports are there in WHO database from the data of the Indian population. And previously, we were on the 10th rank in reporting of the adverse drug reactions, but today the reporting has been increased and this ranking has been shifted to 9th rank, and today India is one of the largest contributors of the individual case safety reports to WHO Uppsala Monitoring Center at Sweden. This slide shows the quality score of the Indian ICSRs being sent to WHO, and here we can see that this blue line, it depicts the quality score of Indian ICSRs, and we can see a lot of difference in the quality score of Indian case safety reports vis-a-vis other countries of the world, and it is a challenging task that we have to keep the quality maintained and to reach to the scale of 1. So it remains a challenging task and an important responsibility for all the stakeholders who are associated with Pharmacovigilance Program of India. For the 4 years, the Pharmacovigilance Program of India had also been given the status for as a WHO collaborative center for pharmacovigilance in public health programs and regulatory services. And now this tenure has been over, but we have again applied for redesignation of this center under as a collaborative center in pharmacovigilance to World Health Organization. And we are expecting that it will be renewed soon. We have the collaboration with various national health programs, such as with the National Tuberculosis Elimination Program, then with the National AIDS Control Program organized by National AIDS Control Organization, NVBDCP, and Universal Immunization Program under the Ministry of Health and Family Welfare, Government of India. And we also collaborate with ICMR and the NABH. Now, during the COVID-19 pandemic, it has been a tough time, and still we have been passing through a tough time. So during this time, the reporting had become difficult. There was a lot of fear in the mind of the officials working in the hospitals. And during this period, now the ICMR had released an advisory, and there was a mention of the Pharmacovigilance Program of India that whenever any adverse drug reaction takes place, then it must be reported. And the advisory on hydroxychloroquine was specifically released, and the IPC had played an important role in collecting of the data and sending it to Indian Council of Medical Research, which was also considered by the ICMR National Task Force. So in the revised advisory which was issued by ICMR, the safety data on hydroxychloroquine generated from Pharmacovigilance Program of India was also considered. And under the chairmanship of Professor Y.K. Gupta and with the involvement of the experts from throughout the country, we had designed a specific adverse event reporting form for drugs being used in COVID-19. And so far, we have received more than 13,000 of the adverse events on account of the hydroxychloroquine alone. And data is being continuously sent to Indian Council of Medical Research as well as to the Office of the Drugs Controller General of India. Now, we sensitize different ADR monitoring centers through the emails, through the posters, and we also organized the virtual training program, and we had trained the Pharmacovigilance associates for specific handling of the adverse events during the COVID-19 vaccination. So all efforts are made on behalf of the National Coordination Center to provide the support to all the adverse drug reaction monitoring centers, and we also encourage them to organize the continuing medical education programs and other workshops, etc. Then we have the different channels of reporting of ADRs, such as the ADR PvPI mobile application is there, which is freely downloadable from Android Play Store, and the adverse events can be directly reported through this channel, and various posters on demand are also sent to the ADR monitoring centers across all the centers. Then we had also come out with the communications in the newspapers in Hindi as well as in English, in Hindustan and Hindustan Times, and sensitization measures are on through different channels to increase the awareness about the pharmacovigilance program of India. However, we face the difficulties during the data collection on account of the restrictions, patient accessibility in different wards, fear in the mind of the officials, increased risk of infection, and the hesitation, etc. And however, we had continued working with respect to providing our services, not only in the area of pharmacovigilance, but also to provide the reference substances to the Indian stakeholders and those who are needy. And we continuously look forward the support from the stakeholders for successful running of the pharmacovigilance program of India. And we also have published the highlights of the drug safety alert as well as the signals in WHO pharmaceutical newsletters, and these drug safety alerts are being disseminated through. Yes, ma'am? Okay, but I'm able to see. So from where I should start, madam? The whole thing. Okay. Carry lost connection. Okay. Okay. So we have sent the communications regarding the drug safety alert and other safety information such as the signals which are generated from Pharmacovigilance Program of India, and so far we have come out with these 7 signals, and we are communicating these signals to Central Drugs Standard Control Organization for taking appropriate action and bringing the changes in patient information leaflet. And we are trying to disseminate the information to enhance the outreach of the Pharmacovigilance Program of India and continuously running the skill development programs and coming out with the resource materials of Pharmacovigilance Program of India, such as the PvPI newsletters, annual performance report, and different guidance document for spontaneous reporting, as well as for reporting of the adverse events by the marketing authorization holders. So next training of the skill development program is due from 13th of September to 17th of September, and the registration is still open and it will remain open till 2nd of September 2021. And here I would also like to bring to your kind notice that we will also be commencing the organizing the National Pharmacovigilance Week in alignment with the World Patient Safety Day from 17th of September 2021, and we look forward for the overwhelming response in dissemination of information and sensitization of the stakeholders. And anyone can enroll by sending the letter of intent to the PvPI, and after examination we give the recognition as the Adverse Drug Reaction Monitoring Center, and there are a lot of examples, a lot of advantages of being an AMC under the PvPI, and all the required information from time to time will be sent from the National Coordination Center for PvPI. And I thank you very much for giving me the opportunity to speak in this important guest lecture. Thank you everyone once again.
Thank you for your valuable time and patience also. I would now invite Dr. Jitendra Verma, CEO of Life Care Innovation, to make us aware on the strategies for the management of fungal infections in COVID-19 patients.
Good afternoon, everyone. I would like to thank the Vice Chancellor, Representative Vipin Puri, Professor Sataj for inviting me here, and Professor Vaike Gupta for creating a situation to let me come here and speak to you. Dr. Gupta went to memory lane about this KGMU. I am a person who was born next door in Ayodhya, but this is the first time I came to KGMU. I left UP very early, exited and went to PGI Chandigarh, and never got a chance to know the colleges of my own home state. To come back to the subject of the day, the incidence of fungal infections is much
high, then it is acknowledged or appreciated. The drugs for treatment of fungal infections are very few. There is a fundamental issue at the origin of the lack of antifungal drugs. The foremost reason for the lack of antifungal drugs is that fungi are eukaryotic organisms, and the host cells are also eukaryotic. So if you find a compound that is working on the fungi, in all possibilities, it would also work on the human cells. So if it is more efficacious drug, it's going to be more toxic drug to they move parallelly. And because of that, you have very few antifungal drugs to count. Okay, you can really count on your fingertips. There are three major, I am skipping the slides mostly because I am told that the time is short. So I will probably take help of a couple of slides, but otherwise I'll try to speak without slides. So there are three groups, mainly the azoles, which is tetraconazole, posaconazole, voriconazole, now isavuconazole, and the echinocandins. There are three echinocandins, anidulafungin, micafungin, and caspofungin. And they all have the main problem, poor efficacy, and drug resistance. Those who follow the literature of antifungal drugs would know that sometimes even before the drug comes to the market, the resistance is seen. Sometimes these azoles and echinocandins. And why it happens? There is a lot of work being done on the origin of drug resistance in antifungal drugs. What happens is that during evolution, these pathogens of today have seen these antifungal drugs because they are all produced by the soil organisms, soil bacteria. So they have seen and they have developed resistance, and that's why they have survived and evolved, and they are in this world today. The only drug, antifungal drug, against which you would not find incidence of drug resistance is amphotericin B. Can I have the presentation? Presentation. Yeah. I will continue to speak on penicillin. So, the problems are difficulties in diagnosis. Confirmed diagnosis is very difficult, and in most places it is impossible to do. When you don't have diagnosis, your requirement is broad spectrum. As I told you, most of the antifungals have narrow spectrum. The true broad spectrum drug is amphotericin B. So one would say that if you have a drug, at least one drug, which is broad spectrum, what's the problem? The problem comes that amphotericin B is extremely toxic drug. Extremely. And most of the time, the toxicity is nephrotoxicity. Why it is so nephrotoxic? Amphotericin B is an extremely hydrophobic drug, and it has the higher affinity for sterols. There are two sterols which are of relevance. Cholesterol, present in the human cell, and a unique lipid constituent of the fungal membrane is ergosterol. Fortunately, the amphotericin B has much higher affinity for ergosterol than it has for cholesterol. Now, kidneys are very rich in cholesterol. The cholesterol content of kidney is higher than other organs. And that is why when amphotericin B comes in contact with kidneys, it binds to the membrane of the kidney. Esterol, the amphotericin B will be bound to the cholesterol of the kidney. It will undergo an rearrangement in the membrane and will create pores. And the cellular material will leak out and the cell will die, and that is what is nephrotoxicity. But the same amphotericin B, when it binds to the fungal cell, it also undergoes rearrangement, creates pores in the fungal cell, and the fungal cell will die. Now, the challenge that you have, can you avoid going amphotericin B to the human organs, tissues, kidneys? If you can do that, then you can convert this drug, which has been nicknamed. It's called amphotic terrible. Because of so much of toxicity, it has a nickname. It's called amphotic terrible. So what you need to do, if you can manage this nephrotoxicity, you can convert this amphotic terrible into amphotic terrific. And that is what the scientists around the world have been trying since 1959, when amphotericin B was first made commercially available. There was a time when it was thought that amphotericin B does not have a future at all. Then came a technology called liposome technology. Liposome technology came in existence in 1964 at Cambridge University. It was developed for the study of biological membrane. It was accidentally, while planning an experiment for immune modulation, the scientists involved were using amphotericin B as an immune modulator, and they were not able to dissolve amphotericin B to use it as the reagent. And they heard that there's a guy called Alec Bangham at Cambridge. He has made something which is called bangosome. Liposomes were first called bangosomes in the honor of Alec Bangham. So these bangosomes you can use and make, dissolve your amphotericin. That was it. And later it evolved into a drug. If you can use it as a reagent, why can't you use it as a drug? Now, it was realized if you can put amphotericin B in lipids and then you give injection, if you give infusion, then because it is bound to the lipids already, it would not have temptation to go and bind to the human cholesterol. And that way it may not cause toxicity. And that is how the first amphotericin B product beyond the conventional amphotericin B was born. It was amphotericin B lipid complex in 1988. But it didn't do the expected job. It was liposome, it was lipids, but it didn't have cholesterol. Cholesterol is a navigator molecule. If you have cholesterol in the liposomes, it navigates amphotericin B away from the human cell. So the first requirement for the targeted delivery of amphotericin B is to have a preparation, an intelligently prepared carrier liposome that has cholesterol in the appropriate amount, in the optimal amount, to navigate cholesterol to the target fungal cell. So the next product that came to the world, it was called liposomal amphotericin B. It is suspended in saline, in dextrose. The liposomal amphotericin B had cholesterol. So it did improve upon the lipid complex. The conventional amphotericin B efficacy was something like 33%, 34%. The lipid complex efficacy was not much high, and even the nephrotoxicity was not significantly lower than conventional amphotericin B. But when liposomal amphotericin B came, where cholesterol was involved, the efficacy went up to something like 76, 77%. 3/4 of the patients were benefited, and the toxicity came down from 66% of conventional amphotericin B to something like 40% to 20% to 27%. So the liposomal amphotericin B has the toxicity of 27% approximately, which is not insignificant. It was at that time in India, the government of India started to work on Atmanirbhar Bharat. They funded, there was a mission program of government of India to develop our own liposomal amphotericin B. They wanted only to make India self-reliant. They didn't have an idea this is going to be a miracle. And under the program, the liposomal amphotericin B was developed in India. And it is the only product which has been subjected to all phases of clinical trials in India. And after that, the question was, who will manufacture it? I am an academic turned entrepreneur. I did my PhD, did my postdoc like everybody does in science. And then I returned to India. I was teaching in the university. I did a job. And then I was approached. Why I was approached? Because today I have 45 years of experience of working on liposomes. I started working on liposomes when they were not called liposomes, 1977. And when I returned to India, at that time, we had already started working using liposome technology to make products for healthcare. So the first product that I had opportunity, in fact, the world had opportunity to work was liposomal malaria vaccine. So when I came back here, having some success somewhere else, which is not a subject of discussion today, it was realized that there is a person who can translate the laboratory research into a utilizable product. So the government of India gave me a grant called PASSER. It's called Program Aimed at Technological Self-Reliance. This grant is given for those missions which are extremely important to the country. They should be of national importance. Under this program, a company was created, and this bridge research, the last mile research, the translational research, was a responsibility given to convert this product, which has already been subjected to clinical trial, into a patient-worthy commercializable product. How this liposomal amphotericin B was different from the liposomal amphotericin B which was already in the market. This liposomal amphotericin B, as I told you, one critical component was cholesterol. So this also had cholesterol. So it was a targeted drug. The second crucial component was that in India, we replaced dextrose by saline. So this is a liposomal amphotericin B in saline. What did we achieve by just changing it from dextrose to saline? It was already well known that if you give saline before administration of amphotericin B, the nephrotoxicity comes down. Why then the world did not use amphotericin saline? Because it is also very well established that in saline, amphotericin B will precipitate. This was a huge limitation which was overcome by Indian scientists. So we made in India liposomal amphotericin B is stabilized as suspension in normal saline. The second change, which is not so major change, which is not a big deal, liposomes have a tendency to fuse. And when they fuse, they sit in the bottle, injection bottle. This is one small liposome, this is one small liposome. When they exert pressure, they fuse and they become one. They become large. So everybody makes lyophilized liposomal amphotericin B. In fact, whatever medicines are made, they are generally lyophilized. We overcame that also. So we first decided that we will use saline, which is not stable. Second, we said we will not lyophilize. We will make suspension in which it is not stable. But we have a stable product. So what saline does, when amphotericin is given, let me go to the picture. You would see that there is distal tubule. So when amphotericin is given, distal tubule is often damaged because of the amphotericin action. And when it is damaged, the reabsorption of it does not happen. When the reabsorption of sodium and potassium does not happen, it is wasted. When it is wasted, then the message goes to the glomerular apparatus to stop filtration. And that stopping of filtration is the stopping kidney function. So prevent the body from getting into sodium potassium deficiency. The mechanism is that stop filtration. Now, when you give saline along with amphotericin B, even if I'm using the word even if your preparation is causing damage to the distal tubule, your blood level of sodium and potassium will not come down because you are giving the saline. If it will not come down, the message will not go to stop filtration. If the message will not go, the filtration will continue. If the filtration will continue, kidney function will continue. So because of that, this is the only nephrosafe amphotericin B of the world today. Yeah. It's called liposomal amphotericin B in saline. I would try to take help of one more slide. Yeah. So you can see here, the first one is liposomal amphotericin B in saline. The second one is liposomal amphotericin B in dextrose. The third one is amphotericin B lipid complex. The fourth one is amphotericin B colloidal dispersion. And the fifth one is amphotericin B lipid suspension. The amphotericin B colloidal dispersion has been withdrawn from the market because of high toxicity. It is not an Indian product. The only first Indian product is the one in saline. The suspension, there is no data available. The lipid complex, I already told you, it has high toxicity and efficacy. So let's compare the two products, two liposomal amphotericin B, which is one in dextrose and one in saline. You would see that the composition is very different. Lipid to drug ratio is very high, 45:1 in one of them and 7:1 in another. This makes a huge difference. When you have more lipids, you have more matrix per liposome, you make more number of liposomes. The same if drug is distributed in larger number of liposomes, there is better biodistribution, and because of their better biodistribution, you have low dose and high efficacy. You achieve both things by making an optimal lipid to drug ratio. And you would see that it is reflected. The dose of the one in saline is 1:3 milligram per kg. You can see that. A lot of people ask me, how come you are also liposomal and they are also liposomal? Why their dose is different? You can see that because of this, the dose of the one in saline is 1:3 milligram per kg, and of the other one is 3 to 10 milligram per kg. In mucor days, it really played havoc. Because the dose was 10 milligram per kg, means 10 bottles of liposomal amphotericin B per day, at least for 3 weeks. You can calculate the money. I am not going to calculate. It was beyond the ability of even affluent Indians to be able to afford. And that is why you have such a high mortality and morbidity. Morbidity, people lost eye. Their face got shaped. Now, if you used a lower dose, it will be much more economical, affordable. You had a long discussion on those tools. And you have high efficacy of more than 90%. Okay. And there is no nephrotoxicity in this one, whereas in the other one, it is up to 27%. Okay. Now, These are the European Society's recommendations for liposomal amphotericin B, where the dose is 5 to 10 milligram per mucormycosis. I had this slide only because this is a new for time, so I thought I'd talk about it. But because of paucity of time, I'll close it here, and anybody can ask me questions. Thank you very much.
Thank you so much for your enlightening words. Now, I would humbly request our Honorable Vice Chancellor, Sir, Lieutenant General Dr. Vipin Puri, to present a memento to our special guest, Professor Y.K. Gupta. I would again request our Honorable Vice Chancellor, Sir, to present a memento to Dr. Jitendra Verma, Sir. I would now request Professor Amit Kumar Sachan, Sir, to present a memento to our Honorable Vice Chancellor, Lieutenant General Dr. Vipin Puri, Sir. Thank you, sir. I will now invite Dr. Anuradha Mishra, Professor in the Department of Pharmacology and Therapeutics, to present the vote of thanks.
Thank you, Dr. Lubna. Dignitaries on the dais, ladies and gentlemen, it is my pleasant privilege to propose a vote of thanks on behalf of the Department of Pharmacology and Therapeutics, King George's Medical University. First and foremost, I would like to thank our Honorable Vice Chancellor, Sir, Lieutenant General Dr. Vipin Puri, for believing in the department proposal and entrusting us with the responsibility to carry out pharmacovigilance and prescription audit in the university. Without his guidance and support, the program would not have seen the light of the day. We are thankful for you for your kind presence, sir. Thank you so much. I would also like to thank Professor Y.K. Gupta, sir, an eminent pharmacologist and brightest alumni of our department. We are grateful that he took out time from his busy schedule to be here with us and providing us with unflinching support to run the program in the university. Thank you for your support and guidance, sir. We are proud of you, sir. I would also like to extend my thanks to our esteemed guest, Dr. Sunil Kumar, Director General Health Services, Ministry of Health and Family Welfare, Government of India. Unfortunately, he couldn't make up to the program. Dr. Jay Prakash, Principal Scientist, Indian Pharmacopoeia Commission. Dr. Pooja Gupta, Coordinator, Pharmacovigilance and Material-Vigilance Center, AIMS, Ministry of Health and Family Welfare, Government of India. It's unfortunate that even she could not make it to the program. Dr. Jitendra Verma, CEO, Life Care Innovation. Thank you all for making this program a successful event. I would also like to thank all the faculty members of the university present online and present here with us. Thank you, electronic and print media personnel, for giving coverage to the event. And last but not the least, I would thank the faculty and residents of the department who have worked tirelessly behind the screen to make this program a great success.